Well, welcome everybody. This is the second webinar that the RACI Pharmaceutical Group in New South Wales has hosted. This one's going to be more on the science of COVID-19. I'm just going to do a quick intro for everybody, just so we can just set the scene for the webinar, and then I'll leave it to our experts to give you all the information. So I'm just going to share my screen a minute. So what we've got, as I say, we're going to talk about the aspects of the science of COVID-19. The structure for today, the format is going to be that, you know, our speakers are going to talk for 20 minutes, 15, 20 minutes, something like that. And then we can have questions as we're going along. And what will happen is we will just use the Q&A function on Zoom to do that. And then after our speakers are finished, we'll open the floor for general Q&A. I do have some questions that people provided before the seminar, and also hopefully people have raised them during it. Please do raise them as we go along. One thing to note, it will be recorded, the webinar today. So please do not worry if you miss anything, or if anybody's not been able to tune into this, it will be recorded. And anybody who registered will get the link so they can see where the recording is. If you haven't used Zoom before, the Q&A function's kind of in the bar at the bottom. You can see it here. We've got it circled, the Q&A function. Please just type your questions in. Or every question that's answered will be visible to everyone. You can ask anonymous questions. You don't have to put your name against it if you don't want to. So moving into the actual seminar. So what we've got is unfortunately, David Beattie had a family emergency today, so cannot talk about the surface side of COVID. So we do apologize for that, but obviously family comes first with these things. So we wish him and his family all the best. We will first have Derek talking, Dr. Derek Lowe, and then we will have Professor Tak Key talking. So I'm just going to do a bit of intro of both the speakers. Dr. Derek Lowe, he's one of the first science bloggers, and his site now receives visits in the tens of thousands daily. He serves on the editorial board of the ACS Medicinal Chemistry Letters and on the advisory board of the Chemical Engineering News. He's worked at Novartis since 2018, and he's held previous positions at Vertex, Bayer, and Shearing Plough. And thank you, Derek, because he's joining us all the way from Boston. So it's evening time for him. So hopefully he's had his dinner. And then Associate Professor Tak Key is a researcher in the School of Physical Sciences at the University of Adelaide, where his group uses steady state and time-resolved spectroscopic techniques to investigate molecular processes. And really we'll be talking about that UV exposure side. So I think that's enough from me. I'm just going to hand over to Derek, who's going to do his presentation for you.
Great. Okay. Let me get my presentation going here. And let's see, share screen. All right.
Looks good, Derek.
Got it. Okay. So thanks very much for having me. It is Thursday evening here, and I have had dinner, so I should be reasonably coherent, or at least as coherent as I generally am. I'm really glad to talk about this topic because it's one that, you know, as a lot of people are interested in. I know that I've been getting a ton of emails, a lot of activity on Twitter, a lot of traffic on the website. And honestly, for the past month or two, I've written about nothing else other than the coronavirus. At some point, I'll start writing about general, you know, chemistry and medchem and drug development again. There's a lot of neat stuff piling up, but for now, it's all coronavirus all the time, playing nothing but your smooth coronavirus favorites that you know and love. So tonight, or in the morning for you guys, medicinal chemistry of the coronavirus. There's a lot to cover here, and I wanted to start by talking about the structure and the proteins of the virus itself. So this illustration should be broadly familiar to people. We're all seeing these graphics of a coronavirus. And what we've got is a structure that's made of not that many parts. That's the thing about viruses. I mean, I don't think most virologists would say that a virus is actually alive. I'm no virologist, but I wouldn't say that they're alive. They are just tiny machines ready to hijack a real living system, namely our cells. And they have very few moving parts. From a medchem and drug discovery standpoint, that has some very good aspects and some very, very bad aspects. The good aspects are that you have a chance to cover everything about the structure and function of the virus itself. The bad news is that you get to the end of that list rather quickly, and not all of these things are, so to speak, druggable. So we'll talk about that as we go on. You see from the graphic here, the red is the infamous spike protein, and that is really a key to the entire virus. We'll see on the coming slides how that is what interacts with the surface of human cells. You also have the envelope protein and the membrane protein, the S protein, the E, and the M. And that is pretty much the structure of the virus. There's another one in there, the nucleocapsid N protein, which is inside the envelope, sort of holding the RNA in place. But that's really not very many structural proteins when you think about a system, but that's all we've got. This is a very similar arrangement to a lot of other coronaviruses. What's not shown on this schematic is a lipid membrane envelope. This is an enveloped virus, like a lot of others, and that is actually what makes it susceptible to ordinary things like soap and water. We don't have that on here, but that's not a protein target. It's very hard to deliberately target a membrane, especially one that's not loaded down with interesting proteins like our own cell membranes are. So if you're going to target the virus, you can either go after something about these structural proteins, or if you look below, there's a list called non-structural proteins. The coronavirus only has 29 proteins total. That's it. Most of them are the non-structural ones and a few accessory proteins. It is really kind of terrifying how these things work. We'll go into that, but you have a tremendous amount of functionality in a very small space. These things bootstrap themselves in an alarming way, and you can tell that we have tens of millions of years of evolution behind this. Brutal evolution. Because think of how quickly a virus replicates. You have such a short life cycle, you have such strong selection pressure with the host immune system trying to stop it at all points. This has been an arms race that's gone on around the clock for who knows how long, 20 million years, 50? These things infect all sorts of mammals and vertebrates. So we'll go on to the next slide and talk about what some of these proteins actually do. This is a schematic of the first part of the viral infection. You can see the virus with its spike proteins coming down and binding to the ACE2 receptor on the surface of the cell. Now, this caused a great deal of confusion earlier in the epidemic because a lot of people have heard of ACE, angiotensin converting enzyme, and it's certainly famous in medicinal chemistry because the ACE inhibitors were some of the first modern design from the ground up type drugs in the late 70s. Problem is that's ACE1. This is ACE2. And for a long time, we didn't even know there was an ACE2. We're still not 100% sure of all its functions. It's involved in hypertension like ACE1 is, but it has different substrates. It does different things. And most importantly, the ACE inhibitors that are in every pharmacy do nothing to ACE2. Totally unreactive to it. It's a different active site entirely. But the coronavirus has found that it can recognize ACE2, and it has evolved for who knows how long, getting better and better and better at taking advantage of it. So the spike protein is what contacts ACE2. If we want to get more specific, there's a part of it called the RBD, the receptor binding domain. One little section of the spike protein is what really contacts ACE2. And if you want to get pickier than that, there's a single loop of the RBD that pokes out with just, a couple of dozen amino acids at most. That is the business end. That is the sharp tip of the spear that the virus is displaying. That's what binds to ACE2. Then an enzyme next door to it, which is labeled here as TMPRSS2, that is a crucial part of the next step. The next step happens to the right. The membrane around the virus fuses with the cell membrane, and that is the problem. At that point, it starts dumping its RNA into the cell. This RNA is taken up by the ribosomal machinery that we are full of in our cells, hijacked, and then it produces a couple of very long, well, fairly long polypeptides. Here's that bootstrapping I was talking about. A lot of viruses use this technique. The first thing they do is they make a very small number, 1, 2, 3, of these long polypeptides. There's something, because one part of the polypeptide folds up into a functional enzyme, which then turns around and starts cleaving the polypeptide into more functional pieces. So this is really an autoloader kind of thing. It's just like as if a box opened up in your living room and a robot arm came out of the box and began to open up the rest of the box and drag out robot pieces that built more robot pieces. It uses some of the proteins in our cells to do this, but the key of it is that it's built to do it to itself. It starts processing and cleaving off into more proteins, which then start going about their business. Now, over on the right, that 1B says endocytosis. That is another possible way for a virus to get into a cell. Endocytosis is, of course, a common function of a cell to bring in large outside entities, say large proteins and the like. We think that the virus is taking advantage of this as well. This may be where the famous hydroxychloroquine is working, but frankly, between you and me, I'm not putting any money on that. I'm not even sure I'm putting money on hydroxychloroquine working at all. We'll get back to that. But this is one possible place it works, because one of the things we do know about chloroquine and hydroxychloroquine is that they can affect endosomes and endocytosis. So that might be where it's working. Now let's go back up to the ACE2 and TMPRSS2, because we've got a couple of things labeled in red there. The TMPRSS2, it says camostat mesylate. I have not heard much about that one recently. Earlier on, there was a lot of hope for that because it was reported to be an inhibitor of that enzyme. And as I mentioned earlier, the coronavirus needs TMPRSS2 to do a particular cleavage step to get this membrane fusion going. Unfortunately, camostat is not a very good drug. It's a serine protease inhibitor, but I'm afraid it's kind of a shotgun of a serine protease inhibitor. Sure, it inhibits TMPRSS2. It also inhibits trypsin. It inhibits elastase. It inhibits, name your favorite serine protease. It probably inhibits it. So it's not much of a drug. I have spoken to colleagues about it who have actually worked with it and a couple of other drugs in that class. And I think the way one of them put it to me is there's a reason it's never been approved outside of Japan where it was developed. It's just not that useful. So I don't know if that's going to help us out very much. Now, the other thing you've got is monoclonal antibodies. And it says convalescent plasma. That could just be polyclonal antibodies. That is going to be something we come back to, because I think that is going to be a very important part of the therapy against this virus. But the idea is simple, that an antibody, whether you've raised it yourself, whether you've gotten it from the plasma of someone else who's been through the infection and raised antibodies, or whether you've gotten it as an injection from someone who has found a good antibody and produced it as a pure protein, the monoclonal. Any of those, this antibody is going to come and stick to the surface of the coronavirus, probably to the spike protein. That's one of the places we know that this can work. And that keeps it from doing this process of binding to ACE2 and then fusing membranes. We're starting to get details of how some of these antibodies actually work. It's pretty intriguing. They often recognize that RBD domain, although one was just reported from the Netherlands that doesn't. It leaves the RBD domain free. And in fact, once it's bound to the coronavirus, it still can bind to ACE2, but it cannot do the next step. Something about the way that antibody binds keeps it from doing the next step of membrane fusion, so that stops it in its tracks too. There's more than one way to deal with this. So that's viral entry, and you can see there's a couple of possible targets there. Could you perhaps block up ACE2? Maybe. We don't know what would happen if you had a really nice, tight ACE2 inhibitor. Would the coronavirus spike protein still be able to recognize it? It might, because it doesn't bind perfectly in the active site. It hits some of the surface of ACE2 as well. We don't have any good tight ACE2 inhibitors to try this with, though, so it's still an open question, I guess. TMPRSS2 we talked about. And beyond that, we're going to have to get into the next part, viral infection. All right, you remember those two polypeptide chains that were at the bottom of this graphic? Here they are being cleaved up into chunks by the main protease, IMPRO, which is the imaginative name that we have given the main protease of the coronavirus. You could imagine inhibiting that. And in fact, there are other antivirals and against other viruses that do that. This is where you have the HIV protease inhibitors. This is where you have the hepatitis C protease inhibitors. There seems to be no reason why you couldn't make a coronavirus main protease inhibitor either. People were actually trying to make that during the earlier SARS epidemic. Problem is, look at how long it took to get HIV protease inhibitors and Hep C inhibitors, years. That's how long it would take you to get this, unfortunately. But if you had a main protease inhibitor, you could try putting it in there. Once these proteins are cleaved into their various non-structural coronavirus proteins, those things disperse through the cell and, well, they do a lot of stuff. They mess around with the interferon response that the cell would be doing. They mess around with the release of cytokines that the cell immune system is starting to do. They also come in and do a more thorough hijacking of the transcriptional machinery, because what the virus is trying to do is make more viral RNA and turn that into more viral proteins. This is where remdesivir comes in. You see it in red there. Remdesivir is something that can mess up the RNA polymerase and of the coronavirus. Remdesivir looks like one of the nucleobases, quite a bit like one. The idea is that you would have some sort of synthetic nucleoside that's close enough to where the RNA polymerase will accept it as something that should be in fresh viral RNA. Looks just fine. But then when it comes time to replicate that and comes time to read it off, it bogs down. So this enzyme has to recognize it. The next enzymes in the process have to choke on it. That's not so easy to do. And particularly in the case of remdesivir, well, all of these nucleosides tend to be a little bit of another shotgun. They are just supposed to make non-functional RNA. And that is why we brought out remdesivir, because it wasn't a coronavirus drug. It was originally developed against Ebola, another RNA virus. Not a coronavirus, but it uses RNA, and it uses RNA polymerase. Then they tried it against SARS, another RNA virus. Then they tried it against MERS, another RNA coronavirus. Here comes another RNA virus pathogen. Let's break out remdesivir. They've also broken out every other nucleoside like this. Ribavirin is one that's always tried. There are a bunch of them. None of them work great, but they can all do a little bit. And that's the biggest, I think, that we can expect from remdesivir, is that it will bog the virus down a little bit. It's not going to be a silver bullet. It's not going to be a cure. None of the nucleosides are cures, but perhaps it could slow things down enough for our own immune system to get a better handle. So that's, as we get in step 4, all the translation and RNA replication. You see this thing is making viral proteins. They're going through the endoplasmic reticulum and the Golgi apparatus. They're getting processed and folded right and glycosylated and everything that a protein needs to do. Unfortunately, because what they need to do is assemble another virus. And you see the packaging step number 5 and then virion release. We don't know much about inhibiting these steps. That's why there are no red arrows with the names of drugs pointing to them. Some of these steps are not understood nearly as well. Even if we could inhibit them, that would be a little bit late in the process. It would certainly be better than nothing, but it would be a lot nicer to keep the virus from even getting into the cell, rather than waiting until it's hijacked the entire cell and then stopping things then. So, what kinds of therapies do we have then? First off is what we've been talking about so far: repurposed, approved drugs, things that are already given to humans. That's a huge step up because, as it says, you can deploy these things quickly. They're already fit to be given to human beings. Not many drugs are. So it was obvious that when this pathogen hit, we were going to go back to the libraries of existing drugs and screen them all to see if we could get something that worked. That's where things like remdesivir came from. You've seen some others too. Famotidine is one that might be in there. There's several things that might help out. I'm not expecting gigantic advances from any of them because none of them are targeted to the coronavirus. We don't have any drugs targeted to this coronavirus yet. As it says in the 3rd bullet point, if you want new targeted small molecule drugs, you're going to be waiting several years. It takes that long. You're also going to need more than one. I mentioned HIV and Hep C. Both of those are drug cocktails. You have a nucleoside in there. You have a main protease inhibitor. You often have something else too. You want to hit a virus in 2 or 3 mechanisms all at the same time so it can't wiggle out by random mutation and developing resistance. If you want a cocktail of 2 or 3 completely new drugs against this coronavirus, you better be prepared to wait 8 or 10 years. I don't think any of us want to do that. So that's why people went for the repurposed drugs. They're unlikely to be very effective, but they are available now. One exception to that, though, you've probably heard about the cytokine storm problem with this infection. There are some strange things about this coronavirus. A paper just came out in the last day or 2 in Cell. I blogged about it today. It noted that this virus, and it's different from SARS, and it's different from MERS in this respect, this virus seems to be very good at bypassing the innate immune system. There's an interferon response that's kind of a first line of defense against viral infection. This virus is very good at shutting that down. There's not much of an interferon response at all. And that means that things just get rapidly worse until the adaptive immune system kicks in and a massive burst of white cells and cytokines all hit simultaneously. And that's what gets people into trouble. We know something about shutting down cytokine signaling. We have some antibody drugs especially that can do that. So those are in trials right now. They are not antivirals, but they could keep people out of the ICU and off respirators if they can actually shut down the worst damage. Because at that point, the worst damage is not coming from the virus. It's coming from us, our own immune systems going crazy because they've suddenly realized that this coronavirus has gotten the upper hand. So I think that's probably our best shot at existing drugs, is something that can turn down the cytokine storm. All right, if those aren't antivirals, and we don't have any good antivirals right now, what hope do we have? That's that last bullet point. New biologics, monoclonal antibodies, as I mentioned before, something that can stick to the virus, inactivate it, stop it from even getting into the cell, and turn the immune system against it immediately. A monoclonal antibody would be a great therapy to give someone who's very sick. It could also be a preventative, a prophylactic for someone who's not. Antibodies can hang around in the blood for weeks. You might have yourself a temporary vaccine with a really good monoclonal, the sort of thing you would want to give healthcare workers and other frontline people so they will not catch the coronavirus. That's impressive, but vaccines are going to be the ultimate answer. Vaccines, I think, are the only thing that's going to make this epidemic go away. The way that we made smallpox go away. No human being gets smallpox anymore, thank God. Very few ever get polio anymore. We've almost got that one wiped out. We can actually come close to wiping out the coronavirus in humans. It's still going to be in animal reservoirs, but if enough of us have been vaccinated, people aren't going to get it and they aren't going to spread it. How long is all this going to take? Monoclonals, they're going into humans next month. Vaccines are already, some of them are already into phase 1 and some of them are moving into phase 2. That's going to be the key. Phase 1 is just looking for the dosages, making sure the blood levels are about right. Okay, you did raise antibodies, fine. Phase 2 is where, as we at least say in America, where the rubber hits the road. That will be finding out if you can protect someone from getting infected. So we're going to be getting a lot of phase 2 trials over the next few months, throughout the whole summer and into the fall. Some of these are not going to work. A lot of them are probably not going to work. Vaccines fail about as often as small molecules fail in the clinic, about 90%, which is terrifying. But we have an awful lot of vaccines going in with a lot of different mechanisms, a lot of different platforms and technologies. I honestly am fairly optimistic. I think we will get a vaccine. I'm just not sure how long it's going to take. If everything were to go perfectly, the first time you could have a vaccine sometime in the first half of next year, that would break every existing speed record. 4 to 5 years is the current speed record for a new vaccine from a standing start. Monoclonals, the same way. If we could have monoclonals by the end of the year, that will break every existing speed record. But I think it's possible. It's going to be a significant scientific challenge, an engineering challenge, a manufacturing challenge, and a logistic challenge. On the other hand, we really want this, and we are really ready to throw insane amounts of money, time, and effort at it. I think we'll get there. All right, that's what I've got. And are we going to do questions now? Are we going to move on to Professor Key, or how do you want to do it?
Thank you, Derek. That was wonderful. Glad to see there's a little bit of hope at the end, which is good. I think we can do a couple of specific questions about the talk now, and then we do have 6 or 7 listed, but we might do some of the rest of them afterwards. So there were a couple around that ACE2 in there. Someone asked, is it ACE2 or the ACE2 receptor? They were trying to understand. And what does it normally do, ACE2? Does it convert angiotensin 2 back to angiotensin? Is that correct? Does that make sense to you?
Yes, it does. The nomenclature here is a total mess.
Okay.
And I blame the biologists. As a chemist, I blame the biologists for most everything. But I blame the biologists here because it is utterly confusing. You have angiotensin, and then that's the actual peptide that ACE1 acts on. And it cleaves a little piece off of angiotensin. That angiotensin is more properly called angiotensin 2, but Roman numeral 2, not Arabic numeral 2. So this ACE2 doesn't act on angiotensin 2 at all. It acts on the shorter version of angiotensin 2 that ACE1 delivers. So ACE1 comes to angiotensin, cleaves it off, makes this shorter version, then this ACE2 enzyme / receptor works on it and cleaves it into a smaller species. Now, those smaller species are not as well characterized. We know they're involved in blood pressure regulation, but there's still a lot of arguing going on even about whether or not an ACE2 inhibitor drug would even be useful. A lot of people think it would be, but there are some whose theory says, no, it really wouldn't do much. At any rate, we don't have any ACE2 inhibitors. So to make things even worse, there are also receptors for that angiotensin peptide. Those aren't even on this screen because they have nothing to do with the coronavirus. And you have angiotensin receptor blockers. You see this ACE2 and we're calling it an enzyme, we're calling it a receptor. It's a receptor for the virus for sure, but normally we think of it as an enzyme. Even though we say receptor, it has nothing to do with the angiotensin receptor either. Did I mention there are several kinds of angiotensin receptor? No, the nomenclature is a mess. But people were worried for a while though that the angiotensin drugs that people take, the ACE inhibitors or the angiotensin receptor blockers, that those might actually cause more ACE2 protein to be expressed on cells. It looks like it's the opposite. There have been at least 2 very good studies showing that people who are taking these angiotensin-directed drugs actually have better clinical outcomes with coronavirus infection. So a couple of months ago, everyone was frantically wondering if they should stop taking them. Now, everyone's frantically being told, don't stop. They're actually beneficial.
Okay, good. No, excellent, excellent. What we might do, I think, let's move on to let Tak do his talk, and then we can do some more questions afterwards. So, so if you would like to stop sharing your screen, Derek, or Tak, just take over.
I'm dropping it out right now. Here we go.
Hey, Tak, how you going?
Good, good. I will share my screen right now.
Excellent.
You can hear me okay?
Yeah, I certainly can. Yep, sounds perfect. See your screen as well. It looks good.
Fantastic. Okay. Good morning, everyone, and good evening to you, Derek. And what a fascinating talk. And I do have a few questions. I'll save them for later. Okay, so let me give you some introduction to the effectiveness of UV in terms of helping us fight the coronavirus. So the idea here is that, so let me just move my windows around. Okay, so first let me turn on my laser pointer. And great. Okay, so so let me just give you a brief introduction to what I actually do in my lab. So I must admit, I'm a photochemist, a photophysicist, and I don't actually work on coronavirus per se. And but I do use spectroscopy. I do use lasers. I use light to study molecules. And this is my group based at the University of Adelaide. And the latest thing that we're doing here is actually development of a technique that is the UV visible analog of 2D NMR. So it tells us, gives us information about coupling of chromophores or light absorbing units. Okay, so to start the talk today, I think I'll share with you a very similar cartoon that Derek talked about earlier. So what's interesting is I was able to find an image from the NIH of this is an electron micrograph of the novel coronavirus, and you can see that the spike proteins are certainly showing up really well here. So they look rather menacing, I would say. The focus of my talk to you today will be about this part of the virus here, so about the RNA. So in terms of using light to target this virus here, there are the RNA is a clear target, and primarily because they absorb light in a region that is quite widely accessible to us. Now, there are, in addition to using light, obviously the most common way to actually destroy the virus is by using soap. So that actually targets the membrane that is shown here. So soap basically dissolves away the membrane, and once the membrane is not there, you won't be able to find these membrane-bound proteins, and then so the virus loses its ability to target specific sites that Derek talked about earlier, and so the virus then can become inactive. Now that's one way, and alcohol spray is also very good, obviously, and it does pretty much the same thing. So it dissolves away the membrane, and so the structure is no longer intact, and it loses its ability. Now using light, one of the first questions we should ask is, where does the coronavirus absorb in terms of wavelengths of light? And it turns out there are the organic compounds that make up all the components of the virus do absorb light, but they absorb light in a not very specific region, and targeting those may destroy only a very small component of the virus. However, if one targets the RNA of the virus, basically what it does is that it damages the light, in this case, damages the RNA, and basically rendering the virus inactive in the sense that it cannot replicate anymore. Okay? It loses that ability to replicate. So in at the onset of the pandemic, there were a lot of well-meaning people to try to educate the public about the effects of light on coronavirus, and this is an attempt by an astrophysicist. So the aim here is to, first of all, you see here, this is now a cartoon of the coronavirus again. So there's a first mistake. This size here is not 10 nanometers, it's actually 100 nanometers. So the correction was made quite quickly. Now, here we see several different things. So an electron has a size of 0.1 nanometer, that's about right. And then the atom is about 0.5, we know that, the hydrogen atom, for instance. Now, if you look at the coronavirus, that's 100, not 10, by the way, right? And now this astrophysicist draws a sine wave here, indicating that the wavelength is 500 nanometers, implying that the reason viruses don't have color is because they are not big enough to absorb visible light. And the background of the astrophysicist shows up very well here. So obviously this scientist here hasn't had any lectures on photochemistry or photophysics. As we all know, fluorescein with the size of about a nanometer can absorb visible light. So this is unfortunately rubbish. Okay. Now the attempts by astrophysicists in working on ways to combat coronavirus has not resulted in a lot of good outcomes. So this is another one. Not long after that Twitter post, so there was this news coming out of, unfortunately, Australia. So I think astrophysicists maybe should leave fighting coronaviruses to medicinal chemists. Okay, so to first understand the effects of light, we need to look at the genetic makeup of the coronavirus. So the idea here is when we think of nucleic acids, obviously we understand DNA quite well, the base pairs and things like that. So for RNA, which is the genetic material in the novel coronavirus, we have RNA. So the difference between DNA and RNA, first of all, is that RNA is single strand, and then it has uracil instead of thymine in its makeup. So it turns out, yeah, having uracil is an important outcome of actually targeting RNA using UV light, and I'll explain that. So when we look at the absorption spectra of nucleic acids, we get these two plots here. So first of all, I want to draw your attention to DNA. So it turns out they absorb, right? DNA and RNA absorb in very nearly identical regions of the electromagnetic spectrum. Now, this is unsurprising. This is to be expected, and the reason is the chemical differences in terms of the light absorbing units is not so great. So uracil and thymine, they, in terms of light absorbing functions, right? They are quite similar. So if you now look at the region here, so this is, the peak is actually for DNA, the peak is at 260. And we see that the DNA absorption shows a huge dependence on the pH value. So basically at very high pH, the helical structure is broken, so you end up with single strands DNA, and the fact that the base pairing effect is not there actually causes DNA to absorb more light. And this is actually a technique, a spectroscopic technique that many people use to actually understand the melting of DNA. And at very low pH, essentially base pairing is no longer present due to protonation of the bases. So in the end, you also end up with single strands and the same effects, right? An increase in absorption can actually be observed. Now for RNA, there's no such an effect, and the reason is it's already a single strand. But the basic point here is that the absorption of RNA happens around, has a peak at 260 nanometers. And the absorption of UV light, okay, in this region has to do with the bases. So these are the chromophores, okay, for RNA. And essentially, there's absorption of UV light due to the presence of these aromatic groups here. Okay, these are you can see a very clear conjugation here and here as well. Okay, so so the presence of pi electrons is the reason why we have UV absorption of RNA. Now, in the absence of pi electron, basically you have absorption further down in the UV. Okay, so so to look at UV is useful, I think, for us to now look at the different types of UV light. Now, a very important message today is that you can see that the absorption begins to take place around about 300 nanometers, okay? And below which we have an absorbance of close to 0. So meaning the astrophysicist was actually right. Coronavirus doesn't show any color, and the reason is because it doesn't absorb any visible light. It's just she got the basics not quite right there. Now, UV. So if you want to target the RNA of the coronavirus, this region here is in UVC. So there's a little bit of a tail here in the UVB region. However, most of the absorption occurs in the UVC region. Now, UVA ranges from 315 to 400 nanometers. That's useless, okay? Completely useless in terms of specifically targeting RNA. Now, there may be some questions about this, the use of sunlight. So as we can see here, sunlight at ground level is mostly visible light plus UVA, and with very, very little UVB. There's virtually no UVC at sea level, okay? And that's because we have a very, very good protective ozone layer taking out UVC, and UVC is definitely present in solar radiation, but it's just that it doesn't reach sea level, and that's a very good thing. obviously, and without UVC, without the ozone layer, UVC would be present at sea level, and that can cause very, very severe damage to DNA. Now, it's interesting looking at it from the evolutionary point of view, because there is a theory in photochemistry that in early Earth, there was no ozone layer, and DNA and RNA actually act as naturally occurring sunscreen for early life form. And it was only until the ozone layer was formed later on that more complex life form can actually evolve, which is quite interesting. And this theory has some basis on it, and the reason is if we now examine the stability of DNA and RNA in the presence of UVC, it turns out they are remarkably stable. Okay, they actually it is quite difficult to cause damage, and the quantum yield for any kind of photochemical reaction is actually very low, and I'll talk about that later on today. And also, partly, one of the explanations is that the excited state lifetime of nucleic acids is actually very, very short. They're on the order of picoseconds. Okay, and then a pico, let me remind you, is 10 to the minus 12. So extremely short. Okay, and you have naturally occurring dyes such as chlorophyll that would live for nanoseconds. So that's 1,000 times longer than the lifetime of nucleic acids in a photoexcited state. Okay, so UVC is necessary. So that's the short message here. So how do we actually get UVC in the lab, or in this case in the clinical environment? So the most commonly found technique is actually using a lamp. So this is a mercury arc lamp. So basically these are essentially like our fluoro tubes, okay? And it's the fluoro tube without the phosphor coating. So can make it really, really bright by maintaining a very high, a relatively high pressure of mercury vapor in there, and by passing through an electric current, an arc can form, and this arc here can then give us UVC. And this, the peak of this emission here is a mercury atomic emission, and that's at 250 nanometers or so. Okay, and these can actually be quite big. So, for example, you can see this one here is probably around about 1.5 to 1.6 meters tall. Now, in the lab, one can also use LED. So LED light emitting diode can now emit in the UVC region to 260, 250 nanometers are now routinely found in light emitting diodes. Now, in terms of using UV to sterilize an environment to destroy the RNA of a virus, there are several considerations to make. So the first question is, how much light is actually necessary? So as I mentioned before, to actually cause damage in nucleic acids is actually quite difficult. So there's a study here, right here, that is quite specific to COVID-19. And the recommendation here is that at least 1 joule per square centimeter is necessary. And let me tell you that's quite intense, okay? Sunlight on a very sunny day is, in 1 second, is about 1/10 of that. So this is 10 times stronger than sunlight on a very sunny day. So to achieve that, we need to basically think about how much time in terms of exposure that's necessary. So let's say we have a UV lamp that emits at an intensity about 3 times the intensity of sunlight on a sunny day, which is 300 milliwatts per square centimeter here. So 3 seconds would be necessary. Okay, that's not too long, that's not too bad, but this is very, very intense. Okay? And that's pretty much like the intensity you would get around about a few centimeters away from something like this. Okay, not very practical. So if you're meters away from a bright UV light, you're likely to get an intensity around about this value here. So that's why a long exposure on the order of minutes to even hours would be necessary to be effective. Okay, and to measure the intensity of light is actually quite easy these days. So one can simply get a power meter that looks like this, and often the active area of the sensor, I should say, of the power meter is has a cross-sectional area of 1 square centimeter. So whatever you measure here tells you exactly the intensity of light that you get. So you can then measure this value, and then that will determine the amount of exposure time that's necessary based on the guideline that's given here. Okay, now I think in the final part of today's talk with you, I'd like to just mention to you what does UV light actually do to RNA? And this is something actually quite that is quite ubiquitous in the area of photochemistry and photophysics. So photo damage, right? Photochemical reactions. So this is the other topic here. Now, there's a paper published in 2012, and this paper here examines the UV effects of RNA, and the conclusion here is that in seconds, right, there's enough already. There's already enough damage in RNA. Now let's go into a little bit more detail. So what is actually happening here? So these are now uracil bases. So often within an RNA sequence, one would be able to find an area where two pyrimidine units are next to each other. So maybe two cytosine units or two uracil units next to each other. Okay, in this case, UV light can actually activate the bases in such a way that in the excited state, formation of a bond that cross-links two bases can happen. So this looks very, very simple to us, right? This looks like a very simple chemical reaction to many of you probably, but that is sufficient to actually cause damage to DNA, and replication can actually be stopped just by this mechanism. And this is the most accessible reaction that is activated or induced by light. Now there are other types of reactions with a lower yield, i.e. photooxidation of the bases. But those happen at a lower yield. Now, these authors here in this paper, they are actually quite clever. So they designed a series of sequences, and they were able to then put them run an electrophoresis experiment in series, and then they were able to use the photo damage, signatures of photo damage, to actually spell some words here. So that's some kind of pun they're making here. But it is quite clear that UV damage is more probable, or with a higher probability, when we have two pyrimidine bases next to each other. Okay. Now, without UV light, basically, this is a control study. There's no photo damage. Now, the RNA follows the 3-second rule in the sense that if we hit it with sufficient intensity, and this is actually a mistake here. This is actually watts per centimeter, not milli. Okay, so apologize for that. So pretty high intensity, so 0.2 to 0.8 watts per centimeter, which is very, very bright UV light. that's sufficient to cause damage in 3 seconds. So you can see that from 0, 1, 2, and then at 3 is when we actually see a significant level of photo damage. And then basically damages just keeps on occurring as you expose RNA to more and more UV light. So 3 seconds, that's the take-home message here with sufficient intensity. And let me just say to you that this is around about 10 to 20 centimeters away from the light source. Okay, so just to conclude today's talk, exposure of the coronavirus to UVC is effective. Okay, UVB is less effective, but UVA is completely ineffective. Now, UVC can cause photo damage, and a lot of times these photo damages are actually permanent in RNA, particularly in viral RNA. And the reason is because, as Derek pointed out earlier, the structure of the virus, the makeup of the virus is incredibly simple. So there isn't a lot of repair mechanisms within this simple structure to actually control the damages. So a lot of times the photo damages are actually permanent. So intensities of hundreds of milliwatts per square centimeters would be necessary. And with such an intensity, only seconds will be needed to destroy the RNA of coronavirus. With that, I thank you for your attention, and I hand over to Scott.
Thank you, Tak. That was wonderful. Thank you very much. We do have a couple of specific questions for you, so I'll go straight into those. Okay. There was a general question around, so virus killing by light, is that general for all viruses? Or that's all RNA viruses, I'm guessing, is it?
Not necessarily. So viruses can have RNA or DNA, but hitting it with UVC is the critical point there. And I also want to point out, to be clear about this. So hitting viruses with UVC is a brute force technique. So it kills everything. It kills your DNA as well. So do not try to expose yourself to UVC, and that's very bad.
Yeah. Okay. No problems. No problems. What about the effect of light on the membrane lipids? Someone's very interested in membranes. Right. It's their forte.
Right. So it turns out exposing viruses to sunlight has an effect. Now that's interesting, right? Because that seems counterintuitive to what I just told you. Because I just said that there's no absorption in the visible region, and that's UV light. Visible light is completely useless. Now, turns out you must understand that viruses have to attach onto a surface, and sunlight can actually heat up a surface, as we all know. And heating up the surface is the key here, not actually photo damage of the virus itself. So heating up the surface obviously causes water to evaporate, and then the membrane structure, lipid structure, would require water, right? The membrane structure would require water to be intact, and without an intact membrane, then the virus can actually become inactive. Now lipids, yes. In terms of photo damage to the lipids, it's possible. However, it's not the most likely photo damage here. Now, the reason is lipids would absorb further into the UV. So it's entirely possible. However, that would not be as effective as targeting the RNA. Obviously the photo damage that's incurred in the lipids would take a lot longer, and the lipids can still, or I should say the membrane can still be quite intact even with some photo damage. So you're going to have to hit it pretty hard. Whereas for RNA, it's relatively easier.
No, sounds good. Sounds good. Look, on a linked one, you might have already answered this. Really, it talked about UV from sunlight will not really affect COVID-19, but does heat affect the structure of the virus? So you're saying that heat is something that is helping in this.
Correct, and it takes hours.
Yeah. Okay.
Right? It takes hours, and that's why you basically dry up the surface, and then the virus cannot survive anymore.
And I've not heard of this. You might have heard of the Pete Evans light machine. Have you heard of this? Pete Evans has got a light machine. Pete Evans, the chef. I think he's the chef guy, isn't he? If you don't know anything about it, you can't answer about it. He's obviously trying to do something with a light machine to kill off COVID-19. So we'll skip that one. Sorry, Alan. We don't have an answer for that one for you.
I don't watch enough TV.
No, fair enough. Fair enough. Just one more quickly, and then we'll bring Derek back into the fore to answer general questions. So I've heard people saying that the ozone layer of Australia is weaker, therefore a higher UV factor. Is that a reason for Australia having the virus not as bad as other places? Is our UV or our temperature or us being in summer anything to do with us getting off lightly?
To some extent, that's true. I should say the UV part is probably not the case here. So
Yep.
It's true. We have clearer sky, so we have higher UV indexes. That's mostly in the UVA, mostly in the UVA region, and a little bit in the UVB, but never in the UVC. So whatever that we observe in terms of the weather, climate, warm climate helping with the infection is literally heat. So sunlight can heat up the surface, and that would not allow the virus to survive for a very long time. Yeah,
cool. Yeah, it's a bit drastic to fly out to space to try and kill off some virus, isn't it? Really dramatic way to go. What we'll do is I'll stop your sharing there, Tack, so we're on the screen. Are you there, Derek?
Yep, sure.
Tack and Derek, you can both bring up the Q&A area if you don't already have it in front of you. So if there's any specific questions you want to answer, but I'll kind of just work my way down them if I can.
Okay. Yeah, I'll just defer to you. Just take us through an order.
Yeah. Okay. No worries. So we have a question early on that was asked while you were talking. Actually, Derek, it was around with regards to repurposing small molecule drugs that target mammalian or bacterial proteins. Are there structural differences in the proteins in viruses that make this particularly challenging?
Yeah, in general that's true, but the bigger difference is that these drugs were developed for completely different proteins.
Right.
Whether they're human proteins or viral proteins, they're just for a completely different purpose. So that's why I'm more optimistic about the cytokine type drugs, because in that case, it's not quite repurposing. You're using them for the purpose that they were intended for, to shut down IL-6 signaling or TNF-alpha signaling or what have you. They're at least being used for the purpose for which they were designed. It's just we found a different use for that mechanism. But a real from the ground up repurposing, like here's a drug that affects enzyme X, and years later we found that it's good for enzyme Y. You can look a long time for an example of something like that. In fact, arguably there are zero. If you want to talk about something that really went to a completely different purpose from its original one, there are close to zero. In fact, probably the best case, and you're going to really be weirded out by this, the best case is probably thalidomide. Because that was originally a sedative, which is just an awful, awful idea. But it turns out it does a lot of very unusual things to transcription, and that's what it's used for now, for instance, myeloma. But if you want to count thalidomide as a drug, as a repurposed drug, then yeah. Otherwise, not so many examples. It's hard.
Yeah, fair enough. There's a couple regarding vaccines. So along the line of, do you think we'd need, you might not know the answers, do you think we'd need annual boosters or every couple of years? Or and from my own personal point of view, I was thinking about, how do you know how long of vaccines last for in the system, and one around just how do you make it sufficient quantities of it? So yeah, on that side,
those are related, of course, because the ideal vaccine is one that you get one shot and it's good for the rest of your life.
Yeah,
that's not always easy to get to, and we don't really know, because there's so many vaccines being developed with so many different technologies some of these technologies are known to, on average, give more lasting immunity than others, but there are always exceptions. Basically, we just don't know enough immunology to tell you beforehand, especially about the duration of action. The only way we're going to know that is to wait and see how long it lasts. For example, a couple of years ago, we started seeing more measles showing up in Massachusetts because of various misinformed people, I'm trying to be diplomatic, who had decided that they didn't want to give their children the measles vaccine. So by golly, we started getting measles again. My wife was volunteering in a hospital and had her measles titer checked. She was no longer immune to measles. That prompted me to go do the same. I was no longer immune to measles. Our childhood measles vaccinations, the rubella part of the vaccine was still good. We were still both immune to rubella. We were not immune to measles, so I got revaccinated. So we don't know. In the same way, we don't know with people who naturally have recovered from the coronavirus. We're not quite sure yet how immune they are and how long that's going to last. The immunity to SARS, which is rather similar, but not quite, the immunity to SARS seems to have lasted just a few years. Who knows?
And the kind of common flu or the vaccinations for that, or so, the things you get every year, the flu jabs and things are changed every year, technically, aren't they? Or altered, or they guess a little bit which one's going to hit. So
they are. The flu, thank goodness, is a different beast because it does all these fancy recombinations with its surface proteins. So it does a mix and match and comes around different every time. So we do have to try to guess. The coronavirus does not do that. That is good.
At least there's something good news.
Oh my gosh, yeah, that is good news. The coronavirus is mutating a bit, but it's not mutating quickly. And none of the mutations so far seem to have too much significance. I know there's a report about one that might have made it more transmissible, but that's very much up for debate.
All right. I'm not sure whether this is both of you or one of you. Someone's asked about vitamin D, and whether it has any effect on the virus or what's been shown to do with vitamin D.
Yeah, I can try fielding that one. There have been a couple of reports on this. It's tricky, though, because they're looking at people with deficiency of vitamin D. There's a lot of, I guess, what you'd call confounding data there, because if someone has a deficiency of vitamin D, they have a number of other things wrong with them, too. So it makes it very hard to tell if this is a cause or an effect. So there's a possibility that there's something there, but it remains unproven, and it's actually going to be pretty hard to prove. The other problem is if, in fact, a deficiency of the vitamin is a bad sign for being infected, it doesn't necessarily mean that taking extra will be a good sign. And the trouble there is that vitamin D is one of the vitamins, vitamin D and vitamin A, where you actually can take too much. You can overdose on those two. Yeah,
fair enough. Is vitamin D come across any of your stuff you've been doing, TAC, at all, or?
Well, vitamin D is naturally generated by exposure to sunlight, so that's a fact. However, whether or not that's correlated to a warmer or sunnier climate, to less occurrences of COVID-19, I think that's unclear still.
Agreed.
There was a question about the difference between SARS and COVID-19, like from like genetic level or down there. Is there a lot of difference, or?
You know, it depends on from where you're standing, because just in terms of viruses in general, SARS is by far the closest human pathogen to the current coronavirus, by far. But it's still not as close as it could be. There's about 83% homology overall, I think, which for proteins is a fair amount, but not a huge amount. That leaves you with a lot of room for variation. The problem is that homology is not a linear kind of function. There are particular amino acids that if they change, like for example, if you were to do a really strong change right in the middle of the receptor binding domain of the spike protein, something that took a positively charged side chain to a negatively charged side chain, that might completely keep it from being infectious. One amino acid out of the entire protein sequence of all the proteins. Whereas single amino acid changes in other parts of it, or ones that aren't so dramatic, probably do nothing. So you got to be really careful when you're talking about homology numbers.
Yeah, fair enough. Fair enough. There's a question about ivermectin, saying that it works in vitro because in parasites against the nervous system, nervous signaling system. You know,
Yeah.
does it work against? Is that shown to work against viruses, or might work against viruses? Do you know anything about that? Okay.
There have been reports, and no one's quite sure how it might work. That's the thing. There are a number of compounds over the years that seem to possibly have some antiviral activity. For instance, hydroxychloroquine is one, and you can actually demonstrate some of that in vitro. Ivermectin is another one, and no one knows how that would be working. It normally affects ion channels, and a type of ion channel that we as vertebrates don't even have, which is why it's so good against worms and arthropods. Now, if you take very high doses of it, it will start to hit some other ion channels, and especially if it gets into the central nervous system. Normally, our CNS pumps it right back out, but it kills turtles. It kills turtles because their membranes around their brain's blood vessels let it right through. It also will kill collies and Australian shepherd dogs. They have a mutation. The sheep herding dog group have a mutation in a particular membrane protein that lets it through. So it could be active. I'm a little skeptical because the amounts that were originally shown to be active in vitro were quite high, and not the amounts that you would get if you took the drug, but it's actually being tested in the clinic. So I'm going to just sit back and wait for the clinical data.
Yeah, fair enough. Fair enough.
Sorry.
Yeah, carry on, Tat, please.
And Derek, there was a question about, I think it was about the origin of the virus. Could it be actually some kind of, so the question
Any
is like,
human intervention, yeah.
Yeah, that's right. Yeah, so Derek and Scott, do you have thoughts about this?
Oh yeah. That's a real tangled one. When you look at the detailed RNA sequence of the virus, and of course, as you've been hearing from everyone here, we know the RNA sequence inside out and a lot of variations on it. When you look at it, you can say unequivocally, it does not bear signs of human engineering. It really doesn't. It compares very, very closely to several other naturally occurring coronaviruses in bats, in pangolins, etc. So it bears no signs at all of having been altered by human hands. Now, there's the other question of whether or not it may have escaped from a lab. We have no idea. I kind of doubt it, but obviously you can't rule that out. But I think we can rule out the idea of it not being a natural virus. It really, really seems to be. I just can't see it.
Well, I won't pretend to understand how you work all this stuff out, but it's good that we can try and dispel some of the conspiracy theories, you know.
Yeah, not all of them. I mean, there's a lot of things that you do to RNA if you're splicing in new bits and trying to alter it, and you leave handiwork behind. You leave the equivalent of chip marks and paint flecks. There's nothing like that.
Yeah, fair enough. That's a good analogy, I think, for people like me that don't understand the technicalities of it all. So there was the question, Attack, we mentioned this before we started actually. Someone, and I'm not even gonna reword it. Someone asked this before about one for Trump, which was basically, how the hell do you swallow UV light to kill the virus from within? Did you want to even attempt?
Take it away.
Okay, so it turns out this is photodynamic therapy is a very, very well-established technique. Okay, so.
And it's been around for a while, has it?
Oh yeah, it has. And so basically, it's most effective on tissues, on lesions that are on surfaces. So maybe on surfaces that's externally, such as your skin, or even in some cases internally. So externally, it's easy to understand. So dye can be injected into the body, and then basically there's dye in the bloodstream, and then by hitting it with light in specific regions, right? And then that can be quite effective. And skin lesions, skin cancer can be treated
Psoriasis.
this way.
But it's non-discriminate, isn't it? So you have to be careful, obviously, because it's
Absolutely.
killing all the good stuff as well.
Exposure of light, absolutely, yes. Exposure of light is most critical. So you can make it more specific by using specific dyes and then specific wavelengths, right? And so that's the best you can do. So just putting UV light in anywhere near you, particularly UVC, not a good idea. Bad idea.
I've got to second that. I've got to second that. As an organic chemist, the kind of UV light that Tak is talking about, UVC, the longer end of that is something that a chemist is certainly no problem working with. 254 is a very familiar wavelength to organic chemists because we use that to visualize fluorescent plates. As you go further down toward 200, it gets nastier and nastier. And below 200, I've never worked with that. That is serious, hardcore ultraviolet radiation. That will damage your eyes. It will damage your skin. It will carve a path.
Very much so. And I work in a laser lab, and we are, it's interesting that we are not so scared of visible light because we can actually see it. When things happen, we can see it. But with UV light, we're extremely careful. And
that stuff will blind you. And I don't think Professor Tak mentioned it, but below 200, chemists and others tend to call that vacuum UV because you almost have to use it in a vacuum because the air starts absorbing it along the way. And if the air will absorb it, imagine what your eye is absorbing.
Absolutely. Water, particularly. Yeah, water absorbs it, and we're full of water.
Yep. Yeah, look, and you're not lighting your house with that stand-up light you were showing us earlier on your presentation, are you? You know what I mean? You're keeping well away from that for long periods of time. There was a question about do coronaviruses have an equivalent to the integrase catalyzed step in the HIV life cycle? Any ideas on that? Oh, okay. Well, at least you know something about it.
I don't think they do. This is why none of these HIV drugs have been of the slightest use against the coronavirus.
Right.
They tried the combination of lopinavir and ritonavir 2 or 3 times early, and it does nothing. So unfortunately, the HIV drugs and the HIV mechanisms don't have much to teach us. But the good news is that's because the coronavirus also is not targeting the CD4 plus cells that the HIV does. If you know anything about immunology, you understand that that's like tossing a grenade right into things, and the coronavirus doesn't do that. And I am very happy to report that.
Oh, that's good. We did talk about vaccines. Someone asked about, is it difficult for a vaccine that attacks the lungs? Does it matter? Is that a consideration?
That's a really good question because there are several kinds of antibodies, and there's IgA is the mucosal antibody type. There is some question about that, but only good thing I can say is that the people who have recovered from the coronavirus and have raised neutralizing antibodies to it, those are the more traditional IgM type. They're circulating around the blood, and that seems to have done the job. Now, it's an open question as to whether you might get a better response or a faster response by raising mucosal immunity. No one's really sure. So I think that's going to be a very active area of investigation. There's also the tricky part that raising one variety of antibody can actually sort of class switch and start raising the other. We don't know much about that either, but no, that's a solid question, but we don't have a solid answer.
Okay, fair enough. We've answered that one. Let me Give me a second. Do we think there'd be the possibility of countries developing vaccines and then basically not sharing it? Or there's a lot going on. Are people really talking that well as well between, you know, I know the UK's got a trial. I think they've already started in Oxford University. Are they sharing that information with people in Australia and everywhere else? And
Yeah, I think we're going to end up sharing. There's an
let's hope
international
so.
on this, and the U.S. is acting like the U.S. has been acting the last few years. But no, I don't think we're going to see a country holding on to it. We'll see what happens when a country develops one. My hope is that we're not even going to give anyone the temptation to do that. That we'll have 2 or 3 vaccines that all work.
Yeah. Okay. Fair enough. Fair enough. Did you mention hydroxychloroquine earlier?
I did.
Yeah, there's something about toxicity someone's mentioned here about, is it a good idea?
Right. That is the big worry about HCQ, is that when you look over the bulk of the clinical evidence so far, the evidence for it having benefit against the coronavirus is kind of weak. There are a few studies that show benefit. There are a lot that show no benefit. The evidence for it doing harm is unfortunately stronger. And the cardiac arrhythmia is the big one. We call that QT prolongation. It's a cardiologist term for the little trace of a heartbeat. And there are a lot of old drugs that cause QT prolongation. We know what causes that now. We actually assay for that during early stages of drug development. We don't put any drugs on the market anymore that do that. It's only older drugs. And hydroxychloroquine is one of them. There have been a number of studies where they have had to pull people off of the drug because they're starting to show dangerous amounts of this arrhythmia. And sure, people with lupus and rheumatoid arthritis, people who are trying to prevent malaria, take it all the time. They are not sick with the coronavirus. That's a different thing.
And that's because it affects the ion channels. Is that right?
Yeah. Right, exactly. It's a particular ion channel called hERG. And we assay for hERG very early on because we don't want that trouble in drug development. You can carry a drug on like that, but you have to do a ridiculous amount of safety studies, and it better be a really good reason.
Fair enough. Just a bit more light, and not necessarily lighthearted for him, but back to the Pete Evans thing. If you don't know who Pete Evans is, by the way, Derek, he's on television. He's a chef, paleo, all manner of weird and wonderful things, whether you believe or not, whatever. But he had a light machine that he was trying to sell, a biocharger light emitting for $15,000, claiming that you could cure Wuhan coronavirus with it. And the TGA, so our equivalent, the FDA, fined him $25,000 for basically doing that. So, and as Dennis has kindly pointed out, thank you for sharing that, Dennis. He said he should stick to cooking. That's what he's good at. So, fair enough. It's like you're saying about the astrophysicists, you know, and I stick to what you're good at. Don't stick to trying to cure.
Yeah, that was a pretty funny example. I appreciated that.
That was good. Let me have a look. Well, we've only got 5 minutes left. Was there anything you guys wanted to answer or mention or ask each other while I've got you both here? Is there any other comments you'd thought of while the other questions had come in? If not, I'll have a little search down the questions.
I did see one on there talking. They were talking about how could I comment on the enormous amount of research duplication in academia and industry. It is kind of irritating when you look at it from the outside, but not as much of it is duplicated as you think. We're all trying different approaches with different starting materials and with different technologies. Right now, we need that. We need as many shots on goal as we can get. So I'm all for it right now.
And you mentioned before that trying to break speed records, but the difference is, of course, we've never really had something where the whole world's trying to cure it at once, you know, like it just, the sheer resource has just never been, you know, let's say HIV or things like that. You know, unfortunately, these were things in poorer countries and not everybody was trying to do it at the same time, whereas we're all in this together, aren't we? So hopefully that will speed everything up.
This is new.
Derek, I do have a question about the last time the world faced such a widespread pandemic. It was obviously the Spanish flu. Right. So from my reading of the Spanish flu, I really couldn't quite get a good grasp on what actually happens in the end. I wanted to say that it sort of magically went away. Is that just
Yeah.
What are your thoughts on it?
You know, we didn't have antibody tests at the time. I'd be willing to bet what happened was we actually finally achieved that herd immunity that everyone is talking about.
Yeah.
And we achieved it by killing off millions and millions of people around the world. I think that's what happened, is that that particular strain of influenza ran out of people that it could easily infect.
And unfortunately, it's nature's way, isn't it? Nature's way is just survival of the fittest, isn't it? So you
know what? We're sitting here in front of a worldwide computer network, surrounded by goods and services from around the world. The hell with nature's way. Let's do it our way. We're better.
Yeah, indeed. Actually, you'd mentioned when we talked before, Derek, about herd immunity and about some of the percentages of infection rates. Did you want to make a comment on that, about some of those? Was it a study in Spain of general population and also in Indiana? Is that right? You said?
Yeah. The numbers are coming back remarkably consistent in the places that have had really severe epidemics, like in Madrid, not across all Spain, just Madrid.
Yep.
Or just in New York City, you're seeing double-digit seropositive people, like 10 to 20%. Otherwise, we're seeing single-digit, 2, 3, 4, 5%. That is very interesting. So there's not like this huge reservoir of people who got it and didn't know. Not many people have had it. What we don't know is, are all of these people still going to get it or still able to get it? Perhaps it looks like there are some T cells in our body that have become sensitized to coronavirus via the common cold type coronaviruses that might provide a little protection. We don't know. This is just coming out now. If we're going to be really sunny and optimistic, it may be that a good number of people have protection that we don't know about. It's just as likely, though, that there's 95% of these populations that can still get it.
So even in the worst affected places, what's herd immunity? Is it 40, 50%? Is it 60%?
For a virus with this level of infectivity, for its R value, you're probably looking at 60 or 70%.
So from and those studies are well done. They the results are quite reliable on the levels. Yeah, but we're not even close, are we? Even in Wuhan, for example, we're not even close.
Right.
Yeah.
Right. You test healthcare workers in northern Italy, and they're not at 60 and 70%.
Yeah.
No one is.
Okay. Fair enough. That's all good. Well, I think we're at the end. I think that's a decent place to finish. There are more questions. We'll be here all day, and you'll be here all night, Derek. So I appreciate both of your time. That's been wonderful. It's been very insightful. Yeah, thank you very much, and thank you everybody that listened to us. That's great. The recording will be available, and both the presentations, and we'll get that sorted in the next week or so. So
enjoyed doing it.
Thank you again, Derek. Thank you again, Tak. Thank you for your time. It's been wonderful.
Good
Thank you.
Everyone.
Bye now.
See you later.